Welcome to the second video of the Membranes and Cell Compartments unit. The cell membrane is often described as semi-permeable or selectively permeable, which means it allows some molecules to move into and out of the cell freely, other molecules to cross only under certain conditions, and some molecules are never allowed to pass. By controlling what can cross its boundary, the cell membrane functions to separate the internal contents of the cell from the surrounding environment, which allows it to maintain homeostasis, or a constant environment within a narrow window of conditions required to support life. In this video, we will discuss the mechanisms the cell membrane uses to accomplish these functions. The simplest movement into and out of cells is through passive transport, which works by diffusion. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration, without the use of energy. Imagine dropping food coloring into a glass of water as seen here. At first, the food coloring molecules remain concentrated where the drop first landed in the water, but eventually they will spread throughout the water, giving it a slightly blue tinge. The color molecules were at a high concentration in the drop and a low concentration elsewhere in the glass. Those molecules will naturally spread without the use of any energy until they are equally distributed. In this figure, all the molecules have a higher concentration outside the cell and a lower concentration inside the cell. They can diffuse across the membrane into the cytoplasm through either simple diffusion or facilitated diffusion. Simple diffusion occurs when molecules travel directly through the cell membrane. There are very few molecules that can do this because of the hydrophobic interior of the lipid bilayer. So the only molecules that can cross the membrane through simple diffusion are small, nonpolar molecules such as oxygen or carbon dioxide. In fact, this is how gas exchange occurs when you breathe. The level of oxygen in the air is much higher than inside your cells. So when you inhale air into your lungs, the oxygen naturally diffuses through the cell membrane and into your bloodstream. The opposite occurs with the waste product carbon dioxide. The concentration in your cells is higher than the air, so it diffuses out of your cells to be exhaled when you breathe. Molecules that are large or polar cannot move across the lipid bilayer directly, but they can still diffuse across the membrane using facilitated diffusion. During facilitated diffusion, the molecule moves through a membrane protein instead of directly through the phospholipid bilayer. These membrane proteins can either be channel proteins or carrier proteins. Carrier proteins are different than channel proteins because they exist in two shapes, one that is open to the outside of the cell and one that is open to the inside of the cell. When the protein is open and facing the side with the higher concentration, the molecule can diffuse into the opening. Once it enters the protein, it causes a conformational change in the protein, which changes its shape, making it open to the other side of the membrane. Once the protein opens to the other side of the membrane, the molecule is able to diffuse out, causing it to have transported across the lipid bilayer. Carrier proteins will only transport specific molecules that are able to bind to their interior site and cause a conformational change. Channel proteins function like a tunnel through the cell membrane that allow molecules with specific properties, such as the correct size or charge, to travel through the opening. Molecules can travel in either direction based on the concentration gradient. For example, water uses specific channel proteins called aquaporins to enter the cell when the concentration outside is higher than inside, or to exit the cell when the concentration inside is higher than outside. The diffusion of water is specifically called osmosis. Because water can travel into and out of the cell along its concentration gradient, it is important that the concentration of water be similar inside and outside the cell. This similar concentration is called an isotonic solution. If an animal cell is placed in an environment with a higher concentration of water than found inside the cell, it is called a hypotonic solution. The water will enter the cell causing it to swell and possibly burst. If the cell is placed in an environment with a lower concentration of water, called a hypertonic solution, then water will leave the cell and it will shrivel. The cell cannot function properly in a swollen or shriveled state, so it is important to be able to determine if an environment is hypertonic or hypotonic. The difficulty with determining if an environment is hypertonic or hypotonic is that the water concentration is not typically given. Instead, the concentration of solute, not solvent, is provided. For example, if you have a cell that has 85% water, means it's also 15% other stuff. That other stuff is the solute. You take that cell and you put it into a 10% sodium chloride solution. Is that cell going to shrink or is it going to swell? In other words, is this a hypertonic environment or a hypotonic environment? The easiest way to answer this question is to know that the word tonic stands for the stuff or the solute, and the word hyper means high. For example, if you have a little kid that's described as hyper, he has high energy levels. Similarly, hypo means low. For example, someone with hypothermia has body temperature that is too low. So a hypertonic solution will have a higher solute concentration than inside the cell, while a hypotonic solution will have a lower solute concentration than inside the cell. In this example, 10% is less than 15%. So this must be a hypotonic solution. If the environment is 10% sodium chloride, that means it must be 90% water. We can see that water is higher outside the cell than inside the cell, thus water enters the cell, causing it to swell. A hypertonic solution must have a higher solute concentration than the cell. So for example, a 20% sodium chloride solution would be considered hypertonic because 20 is higher than the 15. In this case, it would also be 80% water, and so we can see water would go from the higher concentration inside the cell to outside the cell, causing it to shrivel. Passive transport can only transport molecules if the concentration gradient is in the right direction, from higher on the outside to lower on the inside for nutrients that need to enter the cell, or from higher on the inside to lower on the outside for waste that needs to leave the cell. However, many times the cell needs to move substances from areas of lower concentration to areas of higher concentration, against the concentration gradient. For example, if this cell wanted to transport glucose into the cell to obtain maximum energy, it would need to move the glucose against its concentration gradient. The movement of substances against their concentration gradient requires energy and is known as active transport. There are two types of active transport depending on if the energy source is ATP, the main energy source for the cell, or if it comes from an electrochemical gradient of another molecule. A good example of primary active transport is the sodium-potassium pump. This pump uses energy stored in ATP to move sodium and potassium ions against their concentration gradients. Notice that the protein binds sodium from inside the cell, where it's at a low concentration gradient. Unlike a carrier protein involved in facilitated diffusion, this binding does not change the shape of the protein. Instead, the protein must then use the energy from a molecule of ATP to change its shape, allowing the sodium to be released to the outside of the cell. While in this open conformation, potassium from outside the cell is able to bind before the protein changes back to its original shape. Thus, potassium is then released inside the cell, The sodium-potassium pump moves the two molecules in opposite directions. Sodium exits while potassium enters. The pump is thus called an antiporter. Anti stands for opposite. Some transporter proteins move two molecules in the same direction. These are called symporters.